Our next speaker is Dr. Noelle Noyes. Noelle is an associate professor at the College of Veterinary Medicine here at the University of Minnesota. Noelle completed a DVM PhD program at Colorado State University before joining our faculty in 2018. Noelle studies microbial ecology to improve livestock health and welfare and to minimize the impact of antimicrobial resistance in livestock, human, and populations, and zoonotic pathogens. So with that, Noelle, thank you.
Thanks, Nancy. Thank you. Good morning, everybody. I'm going to move the mic over here so you can see more of me. I'm short. So it's my pleasure to be here to talk to you today on the 50th anniversary of the Leman Conference. I want to use this opportunity to give a very brief history of the past 50 years when it comes to microbiome science and what we know about the microbiome, spend some time talking about what we know today, and what that tells us about how we might use the microbiome in the future in the context of swine and pork production. I was asked to give sort of a futuristic visionary type talk, and I'm going to start it off with a definition. So sorry, Monsi, whoops. Might not be the most visionary way to start a talk, but this word engineering I chose very intentionally in the title because I think it's important. It's an important mindset to have when we're talking about the microbiome within swine production. So the definition I want you all to keep in mind as we're talking today is that engineering is a systematic approach to designing systems and processes to meet human needs and wants. In the context of this talk, the system that I'd like you to think about is the microbiome, and we're going to talk a lot about it in the next 40 minutes or so. The needs are all of the needs that you have in this room. The need to produce healthy pigs, to produce productive pigs, to produce safe food within a safe environment, and of course to do that while making a profit. You all probably also have a lot of wants that I don't have time for today, but we're going to at least talk through all of your needs and keep those front and center when we're talking about this concept of engineering the microbiome. So what is the microbiome? It's actually, I would say, a relatively recent concept. So this is a snapshot of a paper that was published in 1964. And it's fuzzy because you can only get this through interlibrary loan as a photocopy. It's not actually available digitally. And it's a really, I think, important paper because it's the first time that someone had described what we would call today the gut microbiome. I want to read one paragraph from this paper. It actually talks about pigs. So it's titled, "The Bacterial Flora of the Digestive Tract. It is usually assumed that the stomach and small intestine are essentially free of bacteria. We have recently established, however, that the mouth, the esophagus, the stomach, and the various parts of the intestinal tract contain a large, living bacterial population. Most of our studies have been conducted in mice and rats, but preliminary observations indicate that a similar state of affairs exists in hogs." So this is a really new concept because up until this paper in 1964, people had assumed that our insides, the insides of our guts were sterile, largely sterile. And germ theory was fairly predominant, right, where bacteria are bad. And we start to see in the mid-1960s with this type of research, and specifically these two scientists, this realization that actually there's bacteria like all inside of our digestive tract through the whole thing. Today, this concept has morphed into what I would call the concept of the holobiont. So this is a word that's not used very much, but what it means is that when we look at a pig, it's not just a single organism. The pig is actually thousands of organisms. It's the pig itself, and it's all of the microbial cells that inhabit and almost every part of the pig. There are some parts that probably don't have microbes, but most parts of the pig do. And all of these thousands of organisms have to work together in order to promote the normal functioning of the pig. And so actually, the number of microbial cells is equal to or greater than the number of cells, of pig cells in the pig, right? So it's a holobiont. It's not a single organism. It's thousands of organisms that all need to be functioning correctly. Okay, so there's also microbiomes of inanimate things, okay? So when you think about your facility, it's not just the pigs that have a microbiome, it's a microbiome that coats the inside walls of the pipes that bring water to your pigs, right? There's microbiome throughout this entire facility, probably impacting outcomes that you care about, all of those needs that I listed, right? Performance, health, safe food if we think about further on down the line in the pork production process itself. There's actually a microbiome on the food that we eat, and we're starting to do some preliminary studies to know now that actually carcasses in a slaughter plant have a very diverse microbiome. And so this is important because that might be driving things like shelf life, the quality of the pork that's produced when the consumer eats it in their home, and things like food safety, right? So all those needs that we talked about. So the microbiome is extremely ubiquitous. It's just really everywhere. It's in the air that we're breathing. It's all over the place. And the question is, can we engineer those microbiomes, right? Like, they're doing their thing, and they have been for longer than we have been, right? But can we do anything with them and actually put some systematic processes around them to improve those outcomes that we care about, like pig performance, pig health, profitability? So, spoiler alert, we already are, sort of, okay? So I want to talk about how we're already shaping the microbiomes that are relevant to pig and pork production. And the reason we're already sort of engineering the microbiome is because we're engineering things that in turn shape the microbiome, okay? So one big one that you all probably think about is host genotype, okay? So we know that different breeds of pigs, different breeds of mice, different breeds of cattle, you name it, those host genetics do have an impact on the microbiome of the pig. So there's this interplay between genotype and microbiome. And we have been breeding pigs for a very long time, and we continue to do that, and now we have very fine control over that. And in doing that, we're not thinking about it, but we're actually also shaping the microbiome that those pigs then have. Another thing that you engineer is social population structure. What I mean by this is just the people that we hang out with, we sort of swap microbes. We think about it in terms of pathogens, but we swap all types of microbes, right? So when you group pigs and you're commingling them in different ways, different stages of production, you're actually shaping their microbiomes by determining who they're hanging out with in their pen, okay? So that's a big one that we already engineer. It's part of the flow of swine production. Diet. This one's pretty obvious, right? You all have fine control and fine engineering over the diet that pigs receive, and that in turn is shaping their microbiome. The host maturation process. The physiological aging of the pig is also a physiological aging of the microbiome of the pig. And we do things in livestock production to sort of try to tweak this maturation process. Like early weaning is a great example, or late weaning, right? We're sort of trying to speed up or delay the maturation process of the pig. And in doing that, we're probably also speeding up or slowing down how the microbiome is evolving along with the pig. Again, it's a holobiont, right? And finally, environment and management. So anything we do to control the environment, which in pig production, you all are really good at this, right? Like, you've engineered these facilities. It's very impressive to see. All of those environmental controls are shaping the microbiome in the facilities and in the pigs. This is also a really big one in the slaughter plant, right? There's all these interventions that we do for purposes of food safety to knock down pathogens. Those interventions, whether it's high heat, whether it's a lactic acid rinse, those things they are also shaping the microbiome that's on that carcass that then goes further down the line into our food. So you're already making decisions about these 5 things, right? Probably on a daily basis. And in doing that, you're shaping the microbiome. It's just we're missing the engineering piece, right? And that's why the word engineering is so important, because you're probably doing it. You're changing the microbiome right now, but you're not actually thinking about how you're changing it, right? You're doing these other things, and one of the side effects is that the microbiome's changing, but that's been hidden to us. We haven't been able to observe that very well until recently, and so we don't really have a lot of knowledge about how all of these things that we're doing already are shaping the populations of our pigs and of our systems and of our pork products. So how do we get there? Like, what do we need to do to get to this place where we can actually systematically design microbiomes within the livestock production process? Well, I'm going to talk about 4 things today that I think are like key ingredients to getting to this promised land, right? So the first is a detailed understanding of the system's behavior. This is the sort of hardcore science that I love to do. The second is we need really robust tools to be able to do this engineering. The third is a knowledgeable workforce to implement it. And the fourth is that we need team science. So I'm going to go through these for the rest of the talk and try to talk about where we're at today with each of these four key ingredients and do a little bit of crystal ball gazing, which I don't feel super comfortable doing, but I'll try my best to talk about, like, when we're at the 100th Leman conference, like, how may we be engineering microbiomes within swine production. Okay, so a detailed understanding of the system's behavior. So there are some defining characteristics of, like, a swine microbiome. And when I talk about defining the behavior, I mean this in a very systems level way. I don't mean, is there a certain bacteria that's doing this on a certain day? What I mean is, how does the microbiome as a whole behave under certain conditions that we see in swine farms or that we see in pigs? And can we even start to predict that or understand what those behaviors may mean? So these are three characteristics that I feel pretty confident saying that, yes, microbiomes behave this way, okay? So I want to go through, I don't want to define these for you, I actually want to demonstrate to you how they show up in the microbiome of pigs. So the first one is variability. And so I want to demonstrate to you the variability characteristic by first asking you a question. So here's a pig that's newly born. What do we want this piglet's microbiome to look like at birth, at weaning, at market? I actually can't tell you that today. I can't tell you, like, what's the optimal microbiome for this pig, whether it's the gut, the skin, the respiratory tract. And one of the reasons is variability, so I want to talk about this. So a paper came out in 2022, very recently, and in this paper, they wanted to describe the gut microbiome of pigs at different growth stages. Like, what does it look like at different ages, right? And so what they did is they didn't generate any new data. They actually went and they pulled available microbiome data. So there's publicly available microbiome data. They pulled it for 1,192 healthy pigs. So these were pigs that had been described as healthy in this publicly available data between the ages of 0 and 174 days old. And they reanalyzed the data and they looked at, okay, what genera of bacteria are across all of these 1,192 pigs, and they identified 509 unique genera. So a genus would be, for instance, Streptococcus or Lactobacillus or Salmonella, right? So those are three different genera of bacteria, and they identified a total of 509 across all of these samples. They then wanted to describe, okay, well, what are the common ones, right? We've identified 509. Which of these are common and what are they? Because maybe the common ones are important. So I ask you, how many of these genera were found in over 90% of the pigs? Just think about an answer in your head right now. How many do you think are very common out of the 509? And people in my lab who've been told to read this paper can't answer, right? So the answer is 19. Okay, so I think that that's probably lower than a lot of people in the room had in their head. It certainly was when I started doing microbiome science. This is the variability. This is the fact that you can have 1,192 healthy pigs, and they will only share 19 things out of 509, right? So the profile, the microbiome profile of a healthy pig is just different. And so it makes it hard to engineer potentially, and we'll talk about why. Okay, another way to look at variability. So this is a project, we're currently analyzing the data right now, but in this project, which was conceived of and implemented by Dr. Megan Niederwerder at Kansas State, they enrolled 100 piglets at 3 weeks of age, and they randomized them into two different groups. One group got an FMT, so orally they were given a fecal microbiome transplant every day for 7 days, and the other group got a saline, oral saline every day for 7 days. So that's minus 7 through 0 there on the x-axis of this chart, down here. And on day 0, all the pigs, all 100 pigs, were challenged with PRRS virus and porcine circovirus 2. The daily administration stopped at that point, and then they kept sampling, right? So we received these samples. Dr. Yala Dung in my lab, who's a postdoc, processed them and is analyzing the data. And the question is, what does the microbiome profile in the feces look like in these two different groups? I'm actually not going to answer that question today. That'll be for next year's Leman conference. But what I do want to talk about is the variability that we see in these 100 pigs and their fecal microbiome profiles taken over time. And one way we can look at that, so we have 681 fecal samples because we sampled the pigs multiple times. What did the variability look like, okay? So one way we look at that is we do an ordination plot. Each dot on this plot is one of those 681 fecal samples. And dots that are closer together have a more similar microbiome profile. Dots that are farther apart are very different. If all of the fecal samples were exactly the same, these dots would be stacked right up on top of each other, right? But they're not. They're dispersed across this two-dimensional space, indicating variability. So we know some things about these samples that might help us understand why we're seeing this variability, okay? We know the age of the pig when the sample was taken. We know the pen ID, so the housing of the pig. We know the pig's ID, so each unique pig has its own ID. We know the sex, and of course we know the treatment group, whether it got FMT or saline. I ask you again to think about this. Which of these factors, and we're able to do this statistically, which of these 5 factors do you think is probably sort of driving most of the variability that we see in these microbial profiles? Which would be like your top choice? And I'll give you the answer. So it's actually the age of the pig and the pig ID are explaining, sorry for the statisticians in the room, they're explaining about 20%, each about 20% of all of the variability that we see in these microbial profiles, right? So together it's about 40% of the variability we can sort of partition to the age that the sample was taken and the unique pig. The pig is its own little ecosystem, and it has there's a lot of variability pig to pig. Pen ID is 4%, and actually the sex in the FMT treatment accounts for less than 1% of all the variability that we see in these microbial profiles. That doesn't mean it's not important, right? It's just that there's all this noise over that, right? And so that's the variability that we see and that we deal with when we do microbiome studies and when we try to think about engineering microbiomes. So why does it matter? Well, I'm going to give you 3 reasons. The first is that it really blows up our research budgets because we need high sample sizes when you have a very highly variable study system. The second is that what really strikes me when I was thinking about this is that a lot of the efficiencies and a lot of the advancements that have happened in livestock production are because we've been able to reduce sources of variability. Like there's not many things in a pig farm right now that are highly variable, right? We have control and we've tried to reduce that variability because it helps us with more uniform outcomes, right? It helps us more tightly manage things like performance, disease, food safety, pork quality, and things like that. Yet there's this invisible thing within our facilities and within our populations that's very variable between pig to pig and between carcass to carcass. And so is that an area of opportunity? Is that something that might be explaining variability we see in outcomes like response to a vaccine, the efficacy of a feed additive? It's different from pig to pig. Could that be the microbiome, right? So this variability, I think, is probably pretty important and maybe a major area of opportunity. The third reason it's important when we're talking about engineering the microbiome is that it really challenges us to define like what should the engineered microbiome look like. Because again, I have many options of a quote unquote good microbiome. Which one do I engineer towards? It's a good question, right? And it's hard to answer because there's so much variability. So these are just some questions that you have to think about if you're starting to try to design a system. Okay, so I talked about variability. Now I want to get to these next 2 terms, again, by demonstrating to you what they look like. So to start off, I want to talk about another story, not of 100 pigs this time, but 108 pigs. This study was funded by the Pork Checkoff, and we did this in collaboration with Pipestone. Dr. Carissa Odlin, who's now at Holstone, she did this study as part of her master's degree, and Dr. Tara Gare, who's a postdoc in my lab, helped analyze the data. This has been published in 2022, the first part of it. And in this study, I'm going to describe it really briefly to you. We enrolled 108 piglets from a single high-health sow herd. And at weaning, we brought them to a BSL-2 research facility, and we randomly allocated them to 3 different treatment groups, 3 different rooms in the BSL-2 facility. One room was, we'll call it the control room. It the pigs just lived their life in this BSL-2 room. A couple pigs had to receive some penicillin injections, but other than that, they didn't get any antibiotics, did not get any viral challenge. In the other 2 rooms, we challenged the pigs with PRRS virus, and then in one room, we gave a moderate antibiotic protocol after that PRRS challenge, and in the other room, we gave an intensive antibiotic protocol. And our question, we followed these pigs over time, all the way to market and actually beyond, which I'll talk about. But our question was, how does the fecal microbiome profile of these pens of pigs, how does it change both over time, but also in the context of these different antibiotic treatments, right? Okay, so this is what we call a relative abundance plot. And each very thin bar within this plot, so going like these little thin bars here, each of those is a pooled fecal sample, so a pen level fecal sample from those pigs. And I've grouped them into 3 groups: the minimal group on the left, the moderate antibiotic group in the middle, the intensive on the right. And this is the fecal microbiome profile when the pigs were weaned at 20 days of age. This is what they looked like. And so the colors on this chart are different phyla of bacteria that were in the feces, right? So we see that Firmicutes is down on the bottom, it's teal. There's also a bunch of yellow, that's proteobacteria. You see a lot of variability, right? Like the height of each color is different between each of the samples. That's the variability that we talked about. So this is at 20 days of age. Nothing different has been done to these groups of pigs at this time. They then acclimated for a week in the BSL-2 research facility, and we went back and we took another sample. Okay, and so you can see what that looked like. Again, pigs are they're transitioning to their new diet, their new environment, but other than that, nothing else is being done to them. At 27 days of age, all the pigs got a modified live PRRS vaccine in preparation for the challenge. We went back right before the PRRS challenge, 40 days later. We took another sample just to see what the baseline was, right? Again, these pigs have been treated exactly the same in the same facility, just different rooms. This is what the profile looks like, so you can see this shift over time. That's normal as pigs age. 67 days, we give the PRRS challenge to the middle group and the right-hand group, right? Left-hand group is our control group. And we go back about, a week later, and we take another sample. Interestingly, when we ran statistics here, we did see significant differences between the control group and the 2 groups that got the PRRS virus, but no antibiotics had been given yet to the 2 treatment groups, okay? They had just gotten challenged with PRRS. We let that challenge sort of take hold. Then we started the antibiotics at 75 days, so they had the PRRS challenge for like a week. We started the antibiotics, and in the intensive group that included in-feed and in-water antibiotics, so we waited until all of those antibiotic exposures were done, and then we went back and took another sample. And this is where we would expect to see like major differences in the profile of the fecal microbiome, because antibiotics are sort of like a slash and burn assault on the microbiome. This is what we saw. So actually really no large and significant differences between the 3 groups. We then followed these pigs all the way to market weight, which was at 122 days of age, and this is what the profile looked like at that point, okay? Again, very similar between the groups, and actually less variability, so this is sort of the stable period. Okay, so I want to use this story to talk about resiliency. I think that this shows very clearly that microbiomes are actually really resilient, right? I mean, we whacked the microbiome with drugs that are specifically meant to kill or slow its growth. And in the intensive treatment group, we actually gave 4 different classes. We gave CTC, Denegard, Tylosin, and Ceftiofur. And yet, we didn't really see much of a signal from that. And, we do talk about the fact that, you take antibiotics, what is that doing to your microbiome? If you read the literature, actually most people sort of their microbiomes rebound pretty well after an antibiotic exposure, especially at this sort of high level, this phylum level. There are, of course, potentially minor long-term effects, and of course some people have very unfortunate outcomes, right, like C. difficile as an example. But by and large, we take antibiotics and our microbiomes do just fine. So resiliency can be a really great thing, right, if you think about it, but it also, when you think about trying to engineer something, it can be challenging because you're trying to push a microbiome in one direction and it wants to be over here really badly, right? And so when we think about trying to manipulate these microbiomes, we really need to be thinking, okay, how do we work with the behavior of the system to engineer it in a certain direction in a sustained way, right? Okay, extreme sensitivity, which is the last one I want to talk about. On its surface, I say, how can something be resilient and extremely sensitive at the same time? That doesn't seem to make sense, but it's true. That's how microbiomes are. So I want to focus on like what should be the most boring time point of this whole study, which is the day 67 time point. Again, the pigs have been managed exactly the same facility, same sow farm, randomized. Like there should be no differences between the groups at this point. But actually, this is where we saw the largest difference between the three groups. Why? Like, why? Our hypothesis, and I think it's a fairly strong one, is that each room in this BSL2 facility sort of was its own little ecosystem, right? We brought these pigs in when they were weaned, and they had these very variable microbiomes, and things that we didn't measure in this study just sort of like nudged these three microbiomes in like slightly different directions, right? And so by the time you get to day 67, you're actually seeing a structuring, it's social population structure in a way. You're seeing a structuring of the microbiome where it's like just a little bit unique to each room. This sort of is sad in terms of our study because now we have differences between our treatment groups before we've given the treatment. But again, if you start thinking about engineering something and it's extremely sensitive, how much control can we have, right? Like how much can we control? And like if we're trying to engineer something and there's all of this stuff going on in the environment or whatever it is that we don't have control over, that may be like sort of conflicting with our engineering endeavor, right? So something to think about. It gets even more variable in the slaughter plant and more challenging. I'm going to quickly talk through this. This is the second part of the study. So we followed these 108 pigs to the slaughter plant. And in collaboration with QPP and Hormel in Austin, Minnesota, we sampled these pigs. So this was just published, and we looked at their microbiome. We looked at their skin microbiome when they arrived at lairage, and then also on the shackle chain after they had been stunned. We took skin samples, and then we followed them through the line. We took pre- and post-evisceration carcass swabs, and then we also took swabs in the cooler once the carcasses had reached the cooler. And we wanted to know like what the microbiome profile looked like across these things. So this, again, is another ordination plot. And dots that are closer together have a more similar microbiome than those that are farther apart. And each dot is a sample from the skin or the carcass of these pigs. The cluster on the right-hand side, which is the red and the blue, that's the skin samples. Those are actually highly variable, but it's all relative because when we move further down the line into pre- and post-evisceration, which are the black and the orange, there's huge amounts of variability between the carcass profiles, the microbiome of the carcass. And when we move into the cooler, which is the teal dots, it's even larger, right? So this is something that processors, I think, it's very helpful to think about this because, you know, maybe one of these microbial profiles predisposes to shelf life issues. Maybe, you know, package bloat starts here, right? And that describes some of the differences in outcomes we see on the quality side and also the food safety side. All right, so it's going to be hard, but you know, important things usually are. But not all is lost because microbiome science is growing up, and I'm very biased, but I do think that this indeed is the case, and I hope that you'll indulge me a little bit to talk a little bit about a scientist's perspective, my perspective on the state of the science. And the reason it's important is because the maturity of a scientific area, in a way, gives us a sense of like the timeline on the application of the science, right? Like if it's a very young science, it's probably not going to be applied super soon. But if it's at a certain maturity, like we have enough knowledge to start thinking about application. And I think that that's where we are. So I want to just really briefly, if you'll indulge me, give a super brief background to where we are today. So I told you that there was this really important series of studies in the 1960s where they were describing a microbiome. They weren't calling it that, but it was. But we don't really see an explosion of microbiome studies until like mid-2000s. So what happened? What happened was we just didn't have the tools. So people were trying to do culture to look at different types of microbes, but culture is very tedious. Not all microbes will grow in the same thing. Not all microbes will grow in anything that we can come up with in the lab. So it's just very tedious, and we also didn't have the skills to like differentiate bacteria from one another at a very fine level, right? So we needed molecular biology really. And so during this period, like there were really important observations being made, but there was pretty limited in what we could say. And so I'm going to say that during this period, microbiome science was gestating. Okay, it had been conceived, but it was just gestating and waiting for the external world to be like ready. And so what happened then, the birth of like today's microbiome science, is that we got the tools to do it. And so this is a very famous chart where on the y-axis, I'm showing you the cost per megabase of DNA sequence, and on the x-axis is the year. And you can see in the mid-2000s, all of a sudden we see like a precipitous drop in the cost that it takes to generate sequence data. That was due to high throughput sequencing technology, and that really allowed us to to be able to generate enough sequence data to describe a microbiome in its completeness with no culture needed. Right? So now we can bypass culture and we can describe these populations based on their DNA. And that was really the birth of microbiome science. And it was only, 20 years ago, right? So it's a new science. And technology is really no longer our bottleneck. You see on that chart, right? We can produce a lot of DNA data like pretty rapidly, pretty cheaply. Now it's really about like, how are we analyzing it? And like, are we analyzing it appropriately? Are we using the right statistics? Are we controlling for confounding? Like all of these fundamental principles of doing good science that sort of like weren't in our heads because we were working in the technology, right? But now we're starting to realize like, okay, actually there have been some really bad misses in microbiome literature, right? It's not no one's fault. It's just like we weren't thinking about these important things because people were trying to just get the technology to work. And so now, a lot of us, I think, feel like this poor drowning older child, right? It's like, oh God, we need to think about all this really technical stuff because we need to be appropriately analyzing all of this data that we're generating, and that's just hard. And so, that's where we're at in terms of like the technology. It's really in the data analysis that we need to be focusing right now, in my opinion. But overall, how mature is the field of microbiome science? Well, I want to so I was a consultant before I went to vet school and got my PhD, and I did consulting for the cell phone, the wireless industry. And we did like innovation strategy and things. So really where the cell phone industry was in like the mid 2000s is sort of like, to me, very parallels where we are with microbiome science right now. And so I went back and looked at some slide decks that we put together specifically for the Sprint and Nextel merger. And this is one of the slides, it's old. But really what we were saying back then is that, Sprint and Nextel, like you have been a technology-centric company. Like you've been focused on like, what is our network? What can it do? Like how big is it? How fast is it? How accurate is it? What's the call quality? And also the phones, right? Like the phones were crazy at this time, right? Like just the different types of phones. But what we were trying to say to them is that like to move forward as an industry, you need to think about customers' needs. And so as a microbiome scientist and we've been very technology centric, like what type of data can we produce, how cheaply, how accurately, how quickly. That's great and we need that, but like we need to move into like you are the customers in this room of microbiome science, in my opinion, as someone who wants to do applied science. What are the needs and how can microbiome science answer some of those needs, right? So I think that that's really where we are. So it's a young field, but it is maturing rapidly, and you're starting to see more people doing microbiome science who do have that applied perspective and are focused on the needs, right? Okay, so knowledgeable workforce is the third one. I don't have any slides for this. I just want to briefly talk about it. If we want to apply microbiome and engineer microbiomes, we need a lot of people who can help with that. And microbiome education is pretty sparse, I would say, so I don't think it's really in undergraduate education much. At the graduate level, there are microbiome classes sort of scattered across different departments right now. So like animal sciences at Minnesota, there's one in computer sciences, ecology, population genetics, microbiology. So they're sort of scattered across like all of the different disciplines that really have to work together to do a microbiome study. But there's no like microbiome major or anything like that. In the veterinary curriculum, it's really interesting. So I graduated in 2018 from vet school, and I don't think we really talked. I mean, I don't remember everything from vet school, I will admit. But I don't think we talked like a lot about the microbiome. We certainly didn't have a class on it, but you could imagine if we generate enough knowledge about this very complex organ system, I'll call it, we may actually end up having like a class like immunology that's called the microbiome. Or maybe it's also sprinkled through other classes, like on the gastrointestinal tract or on the skin. It remains to be seen, but for sure, I think that 50 years from now, you can imagine that veterinary students will get a lot more microbiome exposure. And finally, team science. So I still think that when it comes to applied microbiome research, meaningful data is our Achilles heel. We just don't have a lot of it in the context of microbiome data. What is meaningful? It's things that you all care about. It's like weight data, financial data, disease data, reproductive data, food safety data, food quality data. We have not integrated these data sets very well with microbiome data at this point. And I think that a big reason is because this is the most common question that you see in the scientific literature. It's how do these things over here impact the microbiome, okay? Like the microbiome's the outcome. That is very explainable and understandable because people doing microbiome science up until now have been people who are microbiome scientists, right? They care about the microbiome as its own entity. And so these are the things that you see. I just presented one of my own studies, right, where we looked at antibiotics' impact on microbiome. It's very, very important knowledge. However, probably more useful questions would be this, right, where we flip it and we say, how do different microbiomes impact these things that we care about, right? So we have to flip this a little bit. We have to start gathering this data in the context of microbiome studies in order to answer this. And even more, I think a more impactful way to do it is actually just to make the microbiome like another thing that we're studying in the context of swine production and how all of those things work together to improve or to be risk factors for these important outcomes. And so this is where I hope we can go, but of course this requires really good teams of people, of experts, across all of these different areas, right? And so that's where I hope that this is going, and those are the fun projects to work on, in my opinion. So I hope we are seeing more of those types of studies in the literature, and I think we will see more, a lot more coming up soon here. So, okay, the future. So go back to this newly born piglet. I'm going to just walk you through a scenario of what it might look like to engineer this piglet's microbiome throughout its life. This is purely speculative, so please don't go say, I heard Dr. Noelle Noyes give a keynote and she said that we should, slather the sow's teats with something. Like, I'm not saying that, right? Like, I don't know what this is going to look like, but I'm going to give you a scenario what it might look like. So the way, I love that Pat Horde started his talk with a tree because I'm going to end mine with a tree, okay? So, we didn't coordinate that. So on this tree, you can imagine that a pig is born and it's at the base of this tree. And there are like a million possibilities for what that piglet's microbiome might look like when it reaches market weight. And those are all of the leaves. Millions of possibilities of what that microbiome might look like as it evolves through the course of this pig's life, right? And we might know, because we've done the science, we might know that there's a money branch. This is like a money branch. If you can get the pig's microbiome really early in life, if you can get it down this branch, that pig is highly likely to be a really profitable pig, good outcomes, like resistant to disease, just grows really easy, great. But we know that this piglet, it was born to maybe a sow whose genotype we know doesn't usually produce piglets that have this money tree, this money branch microbiome. And so we're probably not likely to get it down that path. Okay, that's okay. We're gonna engineer this thing, right? We know also that this piglet is actually maybe, so that branch is gone. We can't go down there. There's this other branch early in life, and we know that this piglet might be predisposed for its microbiome to go down that branch, and that's like a no money branch, okay? That's like not good. We want to avoid that branch. So maybe we take these piglets that have like a suboptimal microbiome and are likely to go down a bad path, and we commingle them with some high health piglets. And that social grouping allows them to mix their microbiomes and just sort of nudges them away from this like suboptimal path. So they're going down. We've successfully done that. They're moving up the trunk, and now it's weaning time. And at weaning, we know, okay, like these pigs, again, not super optimal. They're not on the money branch. So we want to support them at weaning. We want to support their microbiomes at weaning specifically, right? And we know that if we delay weaning a couple days and we give maybe like some sort of microbiome cocktail, we will push them towards this leaf over here that we know we want them to go, because that's the most profitable, that's the best leaf on this tree right now that's available to them. We've cut off all these other ones, right? So we nudge them, we do that, we put in some interventions at weaning, we push them down this path. They can't go back, so all these other options are out of the picture, right? We've nudged them further down the road here. And now they reach a branch where, like they could get diverted up to a less good branch. And so we're gonna do something there. Maybe it's adding something to the diet to like prevent them from going in that direction, their microbiomes. And we're successful in doing that, and they reach market, and they have like this microbiome trajectory that has led them to the microbiome that is the profitable one, right? So that is like a way that you could picture engineering microbiomes, but we need a lot of data to do that. We need a lot of expertise to do that. We need the people to do it. And like that's where the hard work is. That's like the fun part for me. And so, yeah, I'm hoping that 100 years from now, or 50 years from now, we will be talking about those things at Leman and talking about like, you know, what we can do to nudge microbiomes in certain directions. And we know that that's like a good direction and that's a bad direction. So with that, thanks so much for listening, and have a great time at the 50th Leman conference.
Thank you very much. Thank you very much, Noelle. Great talk. I forgot to mention that this is the Pijuan lecture, and Carlos would have loved this because he, as many of you know him, he loved research, and Noelle really exemplifies that. So a couple of questions that came in. Maybe you can elaborate more. How do we know? What's the mechanism, or do we know more about how the microbiome is affected by nutrition? And then a segue to that question is, how well has the nutritional community accepted or adopted the microbiome concept? Is there work to be done on the nutrition side to affect the microbiome?
Great question. So I think nutrition is where we probably have like the most information right now about like the specifics of how the microbiome interacts with nutrients in the gut. So yeah, for sure, the nutrition community has embraced, I think, the microbiome. I think where we have opportunities there is it's been, right now it's like very focused, from what I see, a lot of it's focused on, you know, supplementing with like a certain strain of a bacteria or maybe a cocktail of strains. What we don't think about as much with nutrition is like, how is it impacting the whole profile of the microbiome? And that's really important because we don't know like which components of the microbiome are the really important ones right now, and so if you're just focusing on like one or two specific bacteria, you don't know like what that's doing to the rest of the bacteria in that population. So that's like a gap in our knowledge. Yeah, but I think like that's the major gap, and I think that in terms of nutrition, like there's a whole lot to learn, but it is one of the areas where we have like fine control over what the microbiome might look like, right? Because we have fine control over the diet. Yeah.
So in terms of methods, how to study this, it seems like this microbiome area of science seems to like an application for crying use of quantum computing technology. Are you, or do you know of others that are using this technology to study microbiome?
Yeah, so the compute power. So these data sets, we have about 250 terabytes worth of raw sequence data right now, like at Minnesota in our lab. So these data sets are very large. So computer algorithms are something that I work on in collaboration with computer scientists, and for sure the algorithms will need to continue to evolve. But right now, it's really not the algorithms, I would say, that are the bottleneck. A lot of them are being moved over to GPUs, which is great, which is making things a lot faster. And a lot of people are working on compression algorithms, like to make those data sets smaller, but still be able to analyze them. And so people are working on it, and I feel that area is not going to be a major problem. The bigger problem is really like properly interpreting the data based on the algorithm that's being used. That's the bigger problem. A lot of people sort of like run an algorithm and then try to interpret the output, and the interpretation isn't really supported by the way the algorithm works. Okay, so that's kind of the bigger problem that I see.
Well, thank you, Noelle. There were more questions. Please catch up, Noelle, in the breaks. I would like to give you this to acknowledge it's the visual lectureship. Thank you.
Thank you very much, everybody. Thank you.